Welcome to the deep dive.
All right, let's jump straight into a really important topic for your health. Preventing colorectal cancer.
Hmm.
We've gathered up, a stack of research papers, clinical reviews,
A
guidelines,
fair bit of material, yeah.
and our mission, as always, is to cut through all that detail and pull out the most valuable, actionable insights for you. Think of it as your shortcut to being well-informed.
That's right. Colorectal cancer, it's a significant health challenge globally. But the good news, highlighted in these sources, is that prevention and early detection strategies can make a huge difference.
Okay.
We're going to unpack what the evidence shows. Sometimes it's crystal clear, sometimes, well, it's a little complex, and figure out what's most important for you to know.
Excellent. So when we talk about, putting up defenses against colorectal cancer, these sources make it crystal clear where we need to start.
Screening, early detection.
Exactly. Screening and early detection. It's the foundational piece. Absolutely. Screening is just paramount for reducing both the incidence of CRC, that's the number of new cases, and the mortality, the number of deaths from the disease.
And the mechanism behind why screening works seems pretty straightforward based on what these papers lay out, right?
It is, yeah. The vast majority of colorectal cancers develop from these precancerous growths called polyps.
Polyps.
Specifically adenomatous or serrated types. Screening allows us to find these polyps often years before they might turn cancerous and, crucially, remove them.
Remove them right down.
Or, if cancer has already developed, screening helps catch it at a much earlier stage.
More treatable.
Much more treatable, yes.
So for someone who's considered average risk, what are the general age recommendations? When should you start thinking about screening, according to these guidelines?
Typically, screening is recommended to start somewhere between age 45 or 50.
Okay, 45 or 50.
And continue up to age 75. Now, for individuals between 76 and 85, the decision often becomes more personalized.
Oh.
You look at their overall health, life expectancy, what screening they've had before, that sort of thing.
Got it. Now, it feels like screening is such a powerful tool, but one point raised in the sources is that screening rates in the U.S., well, they aren't as high as they could be.
That's a critical point. There's still a gap there, definitely.
Yeah.
And the sources then lay out the different tools we actually have for screening. Broadly, they fall into two categories.
Okay.
You've got stool-based tests.
Looking for markers in your waist.
Right. Done at home, usually.
Yes, usually at home.
Mm-hmm.
And then you have direct visualization methods where you actually look inside the colon.
Okay, let's dig into those methods then. Starting with the stool-based options, what are the key ones and what should we understand about them?
Well, stool tests are appealing, right? They're non-invasive. You do them at home, privately.
Mm-hmm.
The classic one, used worldwide, is the guaiac-based fecal occult blood test, GFOBT.
GFOBT, okay.
This one involves collecting small samples from 3 separate bowel movements.
Yes, and it requires avoiding certain foods and medications beforehand because they could interfere.
Like red meat.
Exactly. Heme-containing foods, some supplements, it's typically done annually. And large studies have shown that regular GFOBT screening can reduce deaths from CRC over the long term.
Okay. And how good is it at actually, finding trouble? The sources mention sensitivity and specificity.
All right.
What does that mean practically?
Good question. So sensitivity is how good a test is at finding the problem if it's actually there. Fewer misses.
Right.
Specificity is how good it is at not giving you a false alarm if the problem isn't there. Fewer false positives.
Okay, misses versus false alarms.
Exactly. For GFOBT, the sources say it has fair sensitivity, meaning it can miss some cancers or polyps.
Okay.
But good specificity for finding actual CRC. So fewer false alarms for cancer itself.
Gotcha.
Oh, and importantly, the office-based single-sample GFOBT isn't recommended for screening. It needs to be the take-home version with multiple samples.
Right, the take-home kit. Okay, so fair at finding, good at not crying wolf too often. What about the FIT test? That sounds like an improvement.
The fecal immunochemical test, FIT, yes. It's highlighted as generally being more sensitive and more specific for CRC detection than GFOBT, according to meta-analyses.
Better on both counts, then.
Generally, yes. And a big plus for many people, it usually only requires a single stool sample.
Just one.
And no dietary restrictions needed beforehand.
That was convenient.
It is. It's also typically recommended annually and has good sensitivity and excellent specificity for finding CRC.
So a pretty solid option. And then there's the newer one, the one that combines a few things. The FIT DNA test.
Yep.
Cologuard is a brand name people might know.
Right. The multi-targeted stool DNA test, or MTSDNA. It combines the FIT component, looking for blood, with testing for specific altered DNA patterns.
DNA from shed cells.
Exactly, shed from tumors or polyps into the stool. The sources note it has higher sensitivity for detecting existing CRC than FIT alone, around 92%.
That sounds good.
But the trade-off is lower specificity, around 90%. So more false positives than FIT.
Ah, okay. More false alarms.
And a significant limitation pointed out is its poor sensitivity for finding the precancerous polyps, especially the larger ones, 1 centimeter or more. It only catches about 42% of those.
Only 42%. That seems quite low for finding the precursors.
It is a noted limitation. It's typically recommended every 1 to 3 years. Medicare covers it every 3 years currently. And it is considerably more expensive than GFOBT or FIT alone.
Right. So better at finding existing cancer than FIT, maybe, but not great at catching those polyps before they turn bad. And more false alarms, plus the cost.
Okay.
Helpful to understand those trade-offs. Now let's move to the direct visualization methods. These sound more involved, maybe, but potentially more powerful.
They are more involved generally. These procedures allow a clinician to actually visually inspect the inside of the colon.
See what's going on.
Exactly. And the key advantage is if they find a polyp, they can often remove it right then and there.
During the same procedure.
Usually, yes. This directly interrupts that pathway from polyp to cancer.
That polyp removal sounds like a huge benefit. But these procedures aren't risk-free, are they?
Correct. There are potential harms. Side effects from the bowel preparation, which, let's be honest, can be unpleasant.
Yeah, I heard stories.
Risks associated with the sedation that's often used. And then procedure-specific risks, like rarely, perforation of the colon wall or bleeding.
Okay, risks to be aware of. Colonoscopy is probably the most familiar one, especially in the U.S. What's the summary on that from the sources?
Right. Colonoscopy is the most widely used direct visualization method here. It allows for viewing the entire colon.
The whole thing.
Yes. It requires the most thorough bowel preparation and typically involves sais. If everything looks normal and the prep was good, it's generally recommended every 10 years.
Every 10 years. Now, something stood out in the material about the type of studies supporting colonoscopy.
Yes, that's an important nuance they highlight. The sources point out that there are no completed randomized controlled trials. You know, the gold standard study design specifically comparing colonoscopy directly to no screening to prove it reduces CRC-specific deaths.
No RCTs versus nothing. That's surprising.
It is, but its effectiveness is still strongly supported by other types of rigorous research, large prospective cohort studies, for instance.
Following people over time.
Exactly. One huge study followed nearly 90,000 people and found those who had a colonoscopy had a significant reduction in both getting CRC 27% lower incidence and dying from it 68% lower mortality.
Wow, 68% lower mortality. That's substantial, even without that specific RCT type.
It is, and large RCTs comparing colonoscopy to other screening methods are currently underway, so we'll learn more.
Okay, so strong evidence supports its impact, just maybe not in that one specific study design compared to doing nothing. What about the risks you mentioned? How common are they?
The potential serious risks are listed, but again, they're uncommon. Cardiopulmonary issues related to sedation, maybe 2.9%. Perforation, very rare, less than 0.1%.
Very low.
Hemorrhage or bleeding, maybe 0.10, 0.6%. Infection is rare. There's also something called post-polypectomy syndrome after polyp removal, maybe around 2.9%.
Okay, and is it perfect? Does it find everything?
Not quite. The sources also note miss rates. Even with a colonoscopy, polyps or even cancers might be missed sometimes. The miss rate for larger polyps over 10 millimeters is around 2.1%.
Still pretty good for the big ones.
Yes, but for smaller ones, 1-5 millimeters, it's higher, maybe 26%.
Wow.
And for actual cancers, the miss rate is estimated between 2% and 6%.
It's incredibly powerful, especially for prevention via polyp removal, but important to know the potential downsides and that it's not 100% foolproof.
Precisely.
Okay, what about computed tomographic colonography? Virtual colonoscopy.
CTC, yeah. That's a less invasive imaging test using a CT scanner. Creates 3D images of the colon.
Like a scan instead of the scope.
Kind of, yeah. It still requires bowel prep, but usually not sedation. They do inflate the colon with air, though, which can cause cramping for some people.
Okay. How often for CTC?
Typically recommended every 5 years, and it's quite good at finding polyps 1 centimeters or larger. Sensitivity is good, specificity very good, to excellent for those larger polyps.
Any particular things to consider with CTC? Kirks.
Well, one unique aspect is the chance of finding things outside the colon on the scan. extracolonic findings.
Like on the kidneys or liver.
Exactly. This happens in a significant number of scans, maybe up to 70%. But only a small fraction, around 3%, actually need follow-up or treatment.
Still could lead to extra tests and maybe anxiety.
It can, yeah. That's a consideration. Also, if anything suspicious is found inside the colon on the CTC, you'll still need a follow-up traditional colonoscopy to remove it or investigate further.
Right, it's diagnostic, not therapeutic.
Correct. And it does involve some radiation exposure, being a CT scan.
Okay. And the last one mentioned is flexible sigmoidoscopy.
Flexible sigmoidoscopy, right. This one only examines the lower part of the colon, about the distal third.
Just the last section.
Yeah. The advantage is it can often be done in a clinic setting, maybe without sedation, and needs a less extensive bowel prep, sometimes just enemas.
Okay. Frequency?
Typically every 5 or 10 years. Sometimes it's combined with doing an annual FIT test as well.
And does it work?
A large Cochrane review found it lowers CRC mortality by about 28% compared to no screening. So it definitely reduces incidence and mortality, at least in that lower part of the colon it reaches.
Makes sense.
Interestingly, one analysis suggested that other methods might be better for women over 60, perhaps because cancer patterns differ. And, like CTC, if polyps or suspicious areas are found, you'll need a full colonoscopy afterward.
Wow, okay. That's quite the menu of screening options.
Yeah.
Each really does have different trade-offs, doesn't it? How invasive, how often, what part of the colon they see, what they find best, the risks, the cost.
Absolutely. It really drives home the point from the sources that choosing a test is or should be a conversation with your doctor.
Yeah.
It depends on the test characteristics, the benefits, the harms, how easy it is for you to actually complete the test, the cost, and your own preferences. The best test is often said to be the one you're most likely to actually do.
That makes a lot of sense. Okay, so screening is our primary line of defense. Catching problems early, stopping them by removing polyps. But what if we could influence whether those problems ever start? That takes us to diet, nutrition, lifestyle.
Exactly. While screening is absolutely crucial, the sources really emphasize that making changes to what you eat and how you live are incredibly attractive strategies for primary prevention, stopping the cancer from developing in the first place.
But reading through the material, the link between diet and CRC seems, well, complicated, not always a perfectly straight line.
It absolutely is complex. Yeah, the sources acknowledge that findings can sometimes be inconsistent across different studies.
Why is that?
Well, it's likely due to really intricate factors, things like your individual genetics, lifelong environmental exposures, the very long time it takes for cancer to develop latency.
Decades sometimes.
Right. And even differences in the tumors themselves, tumor heterogeneity. It's not one single disease.
Okay. So how do the sources approach this complexity to try and find meaningful insights for us?
They prioritize findings from the most robust types of studies, prospective studies that follow lots of people over long periods, and randomized clinical trials, RCTs, when they exist. But they also lean heavily on understanding the potential biological mechanisms. How might diet actually affect things at a cellular level?
Okay.
They pay special attention to pathways involving inflammation, the immune system, and increasingly, the gut microbiota, those trillions of microbes living inside us.
Right, the microbiome.
And they also know that while doing perfect diet intervention trials is really hard.
Getting people to stick to a specific diet for 20 years.
Tough. Exactly. Compliance, dose, duration, it's challenging. But observational data, when combined with a strong biological understanding, still provides very valuable clues.
Okay, let's dig into some specific nutrients then, based on that approach.
Yeah.
Calcium comes up quite a bit. What's the picture there?
Calcium is discussed with probable evidence for lowering CRC risk.
Probable evidence.
Many cohort studies, those large observational studies, have shown that higher calcium intake is linked to lower risk. And RCTs, looking specifically at calcium supplementation, have shown a reduction in the recurrence of adenomas.
The polyps again?
Yes, particularly the more advanced, riskier ones.
But for preventing the cancer itself, were the findings as clear in those big supplement trials?
Not always, no. Some of the largest trials, like the Women's Health Initiative, were kind of inconclusive for CRC incidence itself.
Why the difference, do they think?
Several reasons are proposed. Maybe the participants in those trials were already getting enough calcium from their diet, so adding more didn't help much. Maybe compliance with taking the supplements wasn't perfect. Or maybe the studies just weren't long enough to see a difference in cancer rates. The sources also talk about how calcium might work. It could bind to potentially harmful fatty acids and bile acids in the gut.
Neutralize them.
Sort of, yeah. It might also suppress excessive cell proliferation, encourage cells to mature properly or die off when they should get apoptosis,
Right.
and maybe even interact with DNA damage repair, signaling pathways, the immune system, the gut barrier. It seems to do a lot, potentially.
And does the effect depend on anything else, like other factors?
Possibly. The association might be stronger for cancers lower down in the distal colon or rectum. There also seem to be potential interactions with your vitamin D levels.
Ah, the calcium vitamin D connection.
Right. Also, how much magnesium you eat, maybe genetic variations you have related to the vitamin D receptor, even whether you take NSAID painkillers like aspirin.
Complex interactions. So what's the key takeaway for calcium then?
Based on the sources, the evidence suggests risk increases when intake is below about 700 to 1,000 milligrams per day.
So aim for at least that much.
Yeah. It's considered reasonable to encourage intake above that level, recognizing that while some big trial data is mixed for cancer incidence, the adenoma data is stronger, there are plausible mechanisms, and other studies support it. Of course, you always have to balance that against any potential risks, like maybe cardiovascular links at very high supplement doses, though that's debated too.
Okay, aiming for sufficient calcium, ideally from food, makes sense. How about vitamin D? Seems like there's always a lot of buzz around that one.
Vitamin D is described as having limited suggestive evidence specifically for CRC prevention based on intervention trials.
Limited suggestive. Okay.
But the sources quickly highlight that the observational and experimental data behind it is actually quite compelling.
So lab studies and population studies look promising.
Yes. Studies consistently show that higher vitamin D levels are associated with a lower risk of adenomas, lower risk of getting CRC, and lower risk of dying from it if you do get it.
What's the biological story? Why might vitamin D be protective?
There's really strong biological plausibility. Vitamin D works through the vitamin D receptor, or VDR, which is present in colon cells, and influences the activity of a huge number of genes, maybe 305% of the human genome.
Wow.
It's involved in processes that slow down cell growth, encourage cells to differentiate properly, trigger apoptosis, reduce inflammation, and even inhibit cancer invasion and the growth of new blood vessels that tumors need.
Anti-inflammatory effects sound key.
They are particularly interesting, yeah, especially how vitamin D might interact with immune cells like lymphocytes found within tumors.
But similar to calcium, the direct cancer prevention trials using supplements haven't been definitive yet.
That's right. Results from existing RCTs where cancer prevention was often a secondary goal, not the main one, hasn't been consistently positive. But again, limitations like study duration, the dose used, maybe the baseline vitamin D levels of participants could all play a role.
So maybe the trials weren't set up perfectly to see the effect.
Possibly. The sources mention there are ongoing large trials like VITAL, specifically designed to look at vitamin D and cancer outcomes, which should give us clearer answers, hopefully.
Okay, so what's the recommendation now?
Based on the compelling data we do have from observational and experimental studies and the strong biological rationale, it's considered reasonable to aim for sufficient vitamin D status. The sources often mention aiming for a plasma level of 25-hydroxyvitamin D, that's 25 OHD, of at least 36 nanograms per milliliter as a potential target, based on current understanding.
Okay, aim for sufficiency there too.
Yeah.
Let's talk about fiber, often linked to gut health, and these sources seem to confirm its importance for CRC.
Yes, absolutely. Fiber has convincing evidence for lowering CRC risk.
Convincing. That sounds strong.
It is. Many studies, though maybe not every single one, show that the more fiber people eat, the lower their risk of colorectal cancer.
How might fiber be working its magic here?
Several ways are proposed. It increases stool bulk and speeds up transit time through the colon.
Moves things along faster.
Right, which dilutes potential carcinogens and reduces the time they spend in contact with the colon lining.
Makes sense.
And crucially, when certain types of fiber are fermented by your gut bacteria, they produce beneficial compounds called short-chain fatty acids.
Like butyrate.
Butyrate is the key one, yeah. It seems to be fuel for healthy colon cells and has protective effects. A meta-analysis looking across many studies found a pretty clear dose response, about a 10% decreased risk of CRC for every additional 10 grams of total dietary fiber consumed per day.
10% per 10 grams. That adds up.
Yeah.
Why might some studies not show this link as clearly?
Well, could be confounding factors. Maybe high fiber eaters also have other healthy habits. Could be differences in the types of fiber people eat. Fiber from cereals might be particularly protective in some studies. Maybe there's a threshold effect where the benefit plateaus at very high intakes. or just errors in measuring how much fiber people actually eat.
Dietary recall is tricky.
Very tricky. Also, studies looking at just giving fiber supplements haven't shown the same consistent benefit as dietary fiber.
Why not supplement?
Maybe compliance issues, maybe the supplements don't replicate the complex matrix effects of fiber in whole foods, maybe the follow-up wasn't long enough.
So, given the strong observational data, the biological plausibility, what's the takeaway for fiber?
The sources conclude that given the clear biological mechanisms, the meta-analysis results, the lack of adverse effects from dietary fiber, and its well-known benefits for other things like heart health and diabetes.
Yeah, fiber's good generally.
Exactly. So recommending a high-fiber diet is considered very reasonable for CRC prevention.
Okay. Moving on to B vitamins and methionine. How do these fit into the CRC picture?
These are essential nutrients involved in processes critical for how our cells handle DNA. Things like DNA methylation chemical tags on DNA and DNA synthesis, making new DNA.
Okay, fundamental stuff.
Very fundamental. Folate, or vitamin B9, is the most studied in this context. Folate deficiency has been linked to potentially harmful changes like genomic hypomethylation and problems making DNA accurately, which could contribute to cancer development.
And what do the observational studies show about folate intake and CRC risk?
They've often shown that higher folate intake is associated with lower CRC and adenoma risk. This link seems particularly strong in people who drink alcohol heavily.
Alcohol depletes folate.
It can interfere with its metabolism, yeah. And also in people with low intake of methionine, another nutrient involved in the same pathway. It's often called one-carbon metabolism.
One-carbon metabolism.
Yep.
Sounds a bit technical. Can you simplify that?
Think of it like a fundamental cellular factory. It uses nutrients like folate, B6, B12, and methionine as building blocks and tools. This factory is crucial for making and repairing your DNA and for controlling which genes are turned on or off via methylation. If you don't have enough building blocks or the tools aren't working right.
The factory messes up.
Right, which can potentially lead to DNA damage, instability, and maybe cancer down the line.
Okay, that helps. Now, I've heard there's some debate or complexity around folate, especially with folic acid fortification in foods.
Yes, that's a complex area the sources definitely discuss. There's this dual modulator hypothesis.
Dual modulator.
The idea is that folate might be protective in healthy tissues by ensuring proper DNA synthesis and repair, but it could potentially promote the growth of pre-existing, maybe undetected small cancers or polyps by providing the building blocks they need to make DNA rapidly and grow faster.
So good for normal cells, but maybe fuel for abnormal ones.
That's the concern that fueled the debate around adding folic acid, the synthetic form, to grains. And interestingly, RCTs using folic acid supplements in patients who already had adenomas have mostly shown no benefit in preventing recurrence, or in some cases, even a potential increase in advanced lesions.
Oh, really? An increase?
In some studies, yeah, although some benefit was seen specifically in people who started with very low folate levels.
So the message on folate isn't simply take more supplements.
Definitely not. The role is complex. The timing of intake might be really important. Getting enough folate early in life or before any precancerous changes occur might be protective, but taking large supplemental doses later on could be different.
Right.
However, the sources generally conclude that ensuring adequate folate intake is important to avoid the increased risk linked specifically to deficiency.
Okay, avoid deficiency. What about other B vitamins?
Vitamin B6 intake and blood levels have been linked to lower CRC risk in a meta-analysis. High methionine intake also seems associated with lower risk in some studies, maybe working through similar pathways and potentially reducing inflammation. For riboflavin B2 and cobalamin B12, the evidence is considered inconclusive right now.
Okay. How about antioxidant nutrients? Vitamin C, E, carotenoids, selenium. They're often promoted for cell protection.
They are. These nutrients help protect our cells from damage caused by reactive oxygen species, oxidative stress. That's a very plausible mechanism for cancer prevention. And some early studies, maybe smaller trials, suggested some benefit, particularly for reducing adenoma recurrence.
But what about the bigger later studies?
Well, later, larger cohort studies and big randomized trials looking specifically at supplementation with single antioxidants like high-dose vitamin E or beta-carotene have largely yielded null results for CRC prevention. Didn't seem to make a difference.
So the supplement trials were disappointing.
Mostly, yes. For CRC specifically, Selenium seemed a bit confusing in the sources, too.
Yeah, selenium seemed inconsistent.
It is. Some research suggests a U-shaped relationship, meaning risk might be higher if levels are too low or too high, with an optimal range somewhere in the middle.
Not just more is better.
Right. Different large trials had conflicting results. One suggested benefit, another suggested potential harm, especially in men with high baseline levels. Potential sex differences have been discussed, but it really remains inconclusive.
So the bottom line on antioxidant supplements for CRC prevention, based on this.
Based on the evidence presented in these sources, while getting enough antioxidants from a balanced diet is certainly important for overall health, routine supplementation with high doses of single antioxidant vitamins or minerals just for the specific purpose of preventing CRC is unlikely to be effective, especially if you don't already have a deficiency.
Okay, focus on food sources for those.
Yeah.
Let's talk about fats. There's been this long-standing idea, hasn't there, that high-fat diets might increase CRC risk. What does the current evidence in these sources suggest?
Yeah, that hypothesis has been around a while. The idea was that high-fat diets, especially from animal sources, might increase bile acids in the gut or alter the gut bacteria in ways that promote inflammation, contributing to cancer.
And did the evidence pan out?
Well, some initial studies showed correlations, but subsequent larger studies and meta-analyses looking more closely have found generally no significant association between total fat intake and CRC risk. Same for most specific types of fat, like saturated or monounsaturated.
So total fat, maybe not the main driver.
Doesn't seem to be, based on this evidence. Even a large randomized trial, the Women's Health Initiative again, tested a low-fat dietary pattern and found it didn't reduce CRC risk.
Okay, what about specific types, like omega-3s? They often get singled out?
Yes, omega-3 polyunsaturated fatty acids, the kind found in fatty fish, are highlighted. They have potent anti-inflammatory effects in lab models.
Anti-inflammatory sounds good for cancer prevention.
It does. A small RCT in people with FAP, a genetic condition causing many polyps, showed reduced polyp numbers with omega-3 supplementation. And there's a large RCT called Seafood underway right now looking at omega-3s, aspirin, and polyp recurrence.
So waiting on more results there. What about population studies?
Epidemiologic findings have been kind of inconclusive or inconsistent. Maybe there are interactions with things like aspirin use. Maybe the effect differs depending on where in the colon the cancer is. Maybe it matters when in life you consume them. It's complex. High circulating blood levels of omega-3s have been linked to lower adenoma risk in some studies, though.
Okay, omega-3 is interesting, but maybe not definitive yet. What about omega-6s or trans fats?
Lab studies suggest omega-6s can sometimes promote inflammation, but human studies looking at cancer outcomes, including CRC, haven't really shown clear links. Trans fats, well, they strongly link to heart disease and metabolic problems,
Right.
and they share some biological pathways that could potentially influence cancer. But again, the epidemiological studies looking specifically at trans fat intake and CRC risk are inconsistent, not a clear signal.
So overall, for dietary fats and CRC prevention specifically.
Based on the current data presented in these sources, there's limited clear evidence that specifically altering your total fat intake, or most specific types of fat, is a major strategy for CRC prevention itself. We really need to wait for results from trials like Seafood for more definitive answers on things like omega-3s.
Okay, now this next one was interesting, sulfur. The sources discussed it seeming to have almost opposite effects depending on where it comes from in the diet.
That's right. It's a fascinating area and quite complex. Sulfur in our food comes from various sources. You have inorganic sulfate and sulfur-containing amino acids, particularly from protein sources like red meat.
Okay, sulfur from meat protein.
These can be metabolized by certain gut bacteria into hydrogen sulfide gas.
Hydrogen sulfide, rotten egg smell.
That's the one. Now, H2S actually has important normal functions in the body in small amounts, but excess or chronic exposure in the colon is linked to inflammatory conditions like ulcerative colitis, and it's increasingly implicated directly in CRC development.
So.
High sulfur intake, especially linked to red meat and animal protein, has been associated with flares in UC. And excess H2S might impair the health of colon cells, contribute to DNA damage, promote proliferation, and fuel inflammation.
So sulfur from meat sources, potential problems via H2S, but then sulfur compounds in some vegetables seem good.
Exactly. It's this contrast. Sulfur-containing compounds found in cruciferous vegetables, think broccoli, cabbage, kale.
The brassicas.
Yes. And also in allium vegetables like garlic and onions. These contain different sulfur compounds like glucosinolates and allyl sulfides.
And they do something different.
They get metabolized into different compounds like isothiocyanates, ITCs, from cruciferous veg, and various allyl sulfur compounds from garlic. And these compounds, in experimental models, show anti-cancer effects.
Anti-cancer, how?
They seem to help the body detoxify harmful substances, promote apoptosis, beneficial cell death, modulate inflammation. Basically, the opposite effects of too much H2S.
And is there human evidence for this?
There's some. Modest reductions in CRC risk have been observed with high garlic intake in some population studies, and a meta-analysis found about a 16% reduction in risk associated with higher intake of cruciferous vegetables. Your genetics might even play a role in how well you process these compounds.
So a nutrient with potentially opposing effects based entirely on the food source and the compounds involved.
Precisely. Sulfur from meat potentially contributing to risk via bacterial H2S production, while sulfur compounds from certain vegetables potentially being protective. It's really interesting biology.
But what's the recommendation then?
Well, the sources conclude that while the mechanisms are fascinating, there isn't currently enough consistent human evidence to make a specific recommendation about modifying your overall sulfur intake for CRC prevention. It's more about the food sources, perhaps.
Okay, focus on the foods. Let's step back from individual nutrients then. These sources also look at whole foods and how they fit into overall dietary patterns. Does that give us a clearer picture sometimes?
It often can, yeah. Looking at whole foods and dietary patterns allows us to consider the combined effects of multiple nutrients and compounds working synergistically.
The sum being greater than the parts.
Potentially. And it also accounts for the impact of substituting certain foods for others. If you eat more fish, you probably eat less red meat, for example.
Right. Okay, starting with meat again, but this time specifically red meat and processed meat. We touch on mechanisms like sulfur and heme iron. What's the overall summary on the risk level?
The evidence here is considered quite convincing. Consumption of red meat, and particularly processed meat, increases CRC risk.
Convincing evidence for increased risk.
Yes. The sources indicate a dose-response relationship, higher risk with increased daily intake. This risk seems to rise somewhat linearly, up to maybe about 140 grams per day, then might level off a bit.
And processed meat is worse.
Generally considered higher risk than unprocessed red meat, yes. The association is sometimes noted as being stronger for cancers in the lower parts of the colon, the distal colon, and the rectum.
What about other types of meat? Chicken, fish.
In contrast, poultry and fish consumption are actually associated with a modest reduction in CRC risk compared to eating red meat.
So swapping red meat for chicken or fish seems beneficial.
That's the implication. Based on the convincing evidence presented, it's considered very reasonable to recommend substituting poultry or fish for red and processed meat as a strategy to help reduce CRC risk.
Pretty clear takeaway there. How about dairy products? Milk, yogurt, cheese.
The dairy products, and milk specifically, have probable evidence for protecting against CRC.
Probable protection from milk.
Yes. Higher daily milk intake is linked to lower risk. A meta-analysis found the association might be nonlinear, with the greatest risk reduction seen with intakes around, say, 500 to 800 grams per day, maybe 2 or 3 glasses.
Is that protective effect just about the calcium in milk? We talked about calcium earlier.
Calcium certainly contributes significantly, but the sources suggest other components in dairy might play a role too. Things like conjugated linoleic acid, CLA, butyric acid, especially in butterfat, maybe lactose itself, and the beneficial lactic acid bacteria found in fermented products like yogurt or kefir.
What about yogurt specifically? Is the evidence as strong?
The evidence for yogurt on its own is a bit less consistent across studies, possibly due to variations in the product's live cultures versus heat-treated, sugar content, fat content. It varies a lot.
Okay. And there was that interesting, slightly counterintuitive note about childhood dairy intake.
Right, that was intriguing. One study mentioned suggested that high dairy intake during childhood might be linked to an increased risk of CRC later in adulthood.
Increased risk from childhood intake. Why?
It's not entirely clear. Needs more research, maybe related to growth factors in milk influencing development, maybe effects on the gut microbiome programming early in life. It's speculative at this point, but interesting.
Definitely. But for adult consumption?
For adult consumption, based on the probable evidence for protection, encouraging the intake of milk and potentially yogurt or other fermented dairy is considered reasonable for CRC prevention.
Okay. Fruits and vegetables. They're usually top of the list for any healthy diet advice. But the evidence here for preventing CRC specifically seems less definitive than for, say, fiber or red meat risk.
That's true, yeah. The sources describe the evidence for direct protection against CRC specifically as limited, noting that findings have been inconsistent across different studies.
But they're packed with good stuff, right?
Hmm.
Fiber, vitamins, antioxidants.
Absolutely. Fruits and vegetables contain loads of potentially beneficial compounds, fiber, vitamins C and E, carotenoids, folate, minerals, antioxidants, phytochemicals like glucosinolates in cruciferous veg. The list goes on.
So why the inconsistency in the CRC studies then?
It could be due to several things. The sheer variety of fruits and vegetables makes it hard to study consistently. How they're prepared matters. How much people actually eat varies widely. And it's really difficult to statistically separate the effect of fruits and veg from all the other dietary and lifestyle factors they're often associated with.
The healthy user effect.
Exactly. The association is sometimes stronger for cancers in the lower colon or only seen in specific subgroups of people. Some research suggests there might be a nonlinear relationship. Maybe the benefit for CRC risk plateaus after reaching a moderate intake level, perhaps around 100, 200 grams per day of fruits, and the same for vegetables. More isn't always linearly better for this specific outcome. Also, intake earlier in life might be particularly important, which is hard to capture accurately in studies of adults.
But you mentioned cruciferous vegetables stood out a bit earlier with the sulfur compounds.
Yes, cruciferous vegetables like broccoli, cauliflower, Brussels sprouts do seem to show slightly more consistent potential benefit in some analyses. That meta-analysis suggested about a 16% risk reduction with high intake, possibly tied to those specific compounds like ITCs and how they're metabolized, which can vary based on our genetics too.
Okay. So even if the direct evidence for CRC is less consistent than for some other factors, are fruits and vegetables still a clear recommendation?
Oh, absolutely. The sources stress this point. Even though the direct CRC evidence might be graded as limited or inconsistent compared to, say, red meat or fiber, the well-established, strong benefits of fruits and vegetables for preventing cardiovascular disease, type 2 diabetes, controlling weight, and improving overall health mean that recommending increased intake is definitely reasonable and important, particularly for people who aren't currently eating many.
Right, the overall health package. Got it. And finally, for specific foods, what about whole grains?
Whole grains get a convincing evidence rating for protection against CRC.
Convincing again, like fiber.
Yes, higher intake is consistently associated with lower risk. They are a fantastic source of dietary fiber, of course, but also resistant starch, antioxidants, vitamins, minerals, and various beneficial phytochemicals found in the bran and germ.
Any particular mechanisms highlighted for whole grains beyond just the fiber?
One discussed mechanism relates to their often lower glycemic index compared to refined grains. This is linked to better insulin sensitivity and lower circulating insulin levels over time.
And high insulin is bad for the colon.
Higher insulin levels are thought to potentially promote the growth of colon cells, so keeping insulin lower might be protective. There's also a specific biomarker in blood that reflects whole grain intake, alkylresorcinols, and higher levels of this marker have been linked specifically to lower risk of cancers in the distal colon.
Interesting. So the takeaway for whole grains is clear.
Yes, based on the convincing evidence recommending increased intake of whole grains, choosing brown rice over white, whole wheat bread over white, eating oats, barley, etc., is a reasonable strategy to help reduce CRC risk.
Okay, so we've covered nutrients and specific foods. But looking at the bigger picture, the sources spend time discussing not just individual items, but overall dietary patterns and something called energy balance. Does shifting to that level give us a more powerful message?
It really does, I think. The sources suggest that looking at how foods are combined into overall dietary patterns might reveal stronger associations. It captures those complex interactions between foods and also the effects of substitution. People eating healthy patterns tend to displace less healthy foods.
Makes sense. What kind of patterns are linked to risk in these studies?
A Western pattern diet consistently comes up. This is typically characterized as high in processed meats, red meats, refined grains like white bread, sweets and desserts, high-fat dairy, and sugary drinks. This pattern is pretty consistently linked to increased CRC risk.
The typical modern unhealthy diet.
Pretty much, yeah. On the other hand, a prudent or healthy pattern, usually rich in fruits, vegetables, fish, poultry, legumes, and whole grains, is often, though maybe less consistently than the Western patterns harm, linked to lower risk.
Okay.
Your dietary patterns earlier in life can also matter. And interestingly, sticking to a Western pattern after being diagnosed with CRC is linked to a higher risk of recurrence and mortality.
Wow, so diet matters even after diagnosis.
It seems so. Other defined healthy patterns, like the Mediterranean diet or the DASH diet, dietary approaches to stop hypertension, have also been associated with lower CRC risk in some studies.
And where does energy balance, things like weight gain, obesity, fit into this overall picture? Is it separate from diet patterns?
It's critically linked. The sources highlight that diet strongly influences energy balance, obviously, and being overweight or obese, especially carrying excess fat around the abdomen visceral fat, is considered one of the most important, perhaps the most important, modifiable risk factors for CRC.
Really? Obesity is that significant for this cancer?
Yes, it significantly increases risk, consistently across many studies. The sources explain some potential reasons why. Obesity is often associated with insulin resistance, leading to higher levels of insulin and related growth factors like IGF-1.
And those stimulate growth.
They can stimulate cell growth and inhibit apoptosis in the colon, yes. Fat tissue itself isn't just storage, it's metabolically active. It releases inflammatory molecules called cytokines and other hormones called adipokines that can also promote cancer development. Changes in sex hormones related to obesity might play a role too.
Inflammation from fat tissue.
Exactly. And notice how the components of that harmful Western diet pattern, higher red processed meat, low fruits, veg fiber, whole grains, sugary drinks, are often the very same dietary factors that contribute to weight gain, obesity, and poor metabolic health markers like high insulin. It all ties together.
So the message from looking at patterns and energy balance is really about the whole package working together.
Absolutely. The strong, consistent link between obesity and CRC, and the sources note this link appears particularly strong in men, really drives home the importance of the combined integrated effects of diet and lifestyle. It's not just about single nutrients or foods in isolation, it's about the total amount of energy you consume, energy quantity, and the overall quality of your diet, working together to influence your weight and your metabolic health.
That's a powerful connection. And there's this exciting, relatively new area that seems to tie diet, obesity, inflammation, and our insides together, the gut microbiota.
Yes, the sources definitely emphasize the gut microbiota, those trillions of bacteria, fungi, viruses living in our digestive tract, as a potential key player influencing CRC risk.
Yeah, so
They are constantly interacting with our diet, and they influence us through our metabolism, our immune system development and function, and our inflammatory responses.
and how are these microbes specifically linked to CRC?
Well, studies have found differences. Altered microbial communities, sometimes called dysbiosis, and the presence or abundance of specific types of bacteria, like one called Fusobacterium nucleatum, are associated with both adenomas and cancers in some research.
Certain bugs linked to cancer?
It appears so, yes. And changes in the gut microbiota are also linked to conditions we already know increase CRC risk, like obesity, metabolic syndrome, and type 2 diabetes. It seems interconnected.
And does what we eat change these microbes? Is that the link?
Diet has a profound influence. It's one of the main drivers of the composition and, importantly, the function of your gut microbiota.
Give me an example.
Okay, so a diet high in diverse fibers tends to encourage the growth of bacteria that produce those beneficial short-chain fatty acids, like butyrate, which we said is good for colon health. That's often contrasted with the different types of microbes that tend to thrive on a typical high-fat, high-sugar, low-fiber Western diet, which might produce less beneficial or even harmful metabolites. And the products that our gut bacteria make, those SCFAs, certain vitamins they synthesize, how they modify bile acids that come from our liver, these are really important signals for our own health. If your diet or your microbial community is out of balance, maybe you have reduced levels of these beneficial products or increased levels of potentially harmful ones, and that could contribute to chronic inflammation and disease development in the colon over time.
Wow. It sounds incredibly complex, but really full of potential for future prevention strategies, maybe. Probiotics, prebiotics?
It is definitely an exciting area, but the sources also caution that this field is still relatively early in its research phase. We need many more studies in both animal models and especially well-designed human studies to fully understand the precise mechanisms, cause and effect, and the real implications for cancer prevention strategies like manipulating the microbiome.
Okay, still learning there. We've covered a tremendous amount of ground here. From all the different screening tests and what their results mean, pros and cons, tests to the specific roles of various nutrients like calcium, vitamin D, fiber, folate, the evidence for each.
Right, the individual components.
Then looking at whole foods, the risks from red meat, the benefits from milk and whole grains, the complexity of fruits and vegetables,
Yep, the food level.
the impact of overall dietary patterns, and the huge importance of maintaining a healthy weight.
Energy balance, yeah.
And finally, this emerging connection to our gut microbes. Let's try to bring this all together. What are the key takeaways from this deep dive?
Okay, summarizing the core findings from these sources. While doing perfect long-term randomized trials for dietary interventions and cancer prevention is really challenging,
Yeah, we established that.
consistent evidence from large prospective studies paints a pretty strong picture. Higher intake of red meat, and especially processed meat, is clearly linked to increased CRC risk. That's quite solid.
Okay, limit red processed meat. What about the protective side?
On the flip side, higher intake of dietary fiber, milk, calcium up to a certain level, and whole grains is linked to lower risk. The evidence for these is generally rated as probable or convincing.
Fiber, milk, calcium, whole grains beneficial.
Yes. And when you combine that strong observational evidence with our growing understanding of the biological mechanisms, how these factors might influence our cells, inflammation, our gut environment.
It provides a solid rationale.
Precisely. It provides a strong rationale for pursuing dietary strategies for prevention.
So what are some actionable steps someone could take away from this based on the evidence we reviewed?
Okay, based purely on the evidence presented in these sources, some reasonable steps you could consider include actively increasing your intake of fiber-rich foods, fruits, vegetables, legumes, whole grains,
More fiber.
increasing intake of milk, and possibly other dairy like yogurt. Definitely consuming more whole grains instead of refined grains.
Whole grains over white.
Limiting red meat and especially processed meat, maybe choosing poultry or fish more often instead.
Swap the meat.
Aiming for sufficient calcium and vitamin D intake, ideally focusing on food sources first. Ensuring adequate folate intake to avoid deficiency, but being cautious about high-dose folic acid supplements unless there's a specific reason.
Right, the folate complexity.
And the sources also clarify that, based on current evidence, routine supplementation with high doses of single antioxidants like vitamin E or C or folic acid, or making specific changes to total fat intake or sulfur intake alone, isn't strongly supported just for the specific purpose of CRC prevention.
Okay, supplements maybe not the magic bullet here. And perhaps the single biggest takeaway from the whole diet and lifestyle section. What really stood out?
I think it has to be the emphasis on energy balance. The prevention or reduction of overweight and obesity through a combination of a balanced, healthy dietary pattern and regular physical activity, which we didn't focus on as much but is crucial, is highlighted again and again as a major critical strategy. That link between excess body weight, particularly abdominal obesity, and CRC risk is just so strong and consistent in the evidence.
Maintain a healthy weight is paramount.
It really seems to be, yes.
Okay, it feels like we've learned a lot and distilled some key messages, but the sources also make it clear there are still unanswered questions, right? We don't know everything yet.
Oh, absolutely. Important questions remain, as noted in the material. For example, we need to better understand how specific nutrients or foods might influence different subtypes of CRC differently. Cancer isn't monolithic.
Cancers in different locations or with different genetic mutations.
Exactly. What's the optimal dose or the optimal timing or the best form of a nutrient for maximum protection? How do diet, the gut microbiota, our own genetics, and other host factors all interact dynamically to influence risk over an entire lifetime? These are huge, complex questions still being actively researched.
But it feels like we're making exciting progress in understanding these things.
Absolutely. The sources express optimism. Technological advances, genomics, metabolomics, better microbiome analysis tools are really helping us dissect these complex interactions and move closer to pinpointing causal relationships. That's crucial for refining public health recommendations and ultimately reducing the burden of this really significant disease.
Well, that was a deep dive indeed. Thank you for joining us as we explore this really complex but I think fascinating relationship between what we eat, how we live, how we screen, and the prevention of colorectal cancer, drawing all those insights from the research stack.
It's a powerful reminder, isn't it, of the many factors that are, to some extent, within our influence that can really shape our long-term health trajectory.
Definitely. And as you, our listener, consider everything we've discussed today, maybe here's a final thought to ponder.
Think about how the food on your plate isn't just about calories or basic nutrients anymore. It's actually engaging in this constant, incredibly complex conversation with the trillions of microbes living inside your gut, and how that hidden dialogue, that interplay, might be influencing your health pathway, your risk of diseases like cancer, in ways we're really only just beginning to fully grasp.